Can I start? Yes. Okay. Okay. Good. Uh, uh, Welcome back, and hopefully the coffee has woken you up. That was a, uh much needed, nice break. Thank you, Jonathan, for covering lots of topics that continue from yesterday. Hopefully, then this part will be a little smoother and easier, thanks to all of the heavy lifting in the previous talk. So what I'm going to try to do today is the following. Maybe for the first half of this lecture, we will continue with some of these concepts that we started off yesterday and have continued this morning. And then in the second half of today's talk and then the next talk that I'll give you some real research examples. These are examples from my own work, mostly because I'm familiar with it, not because it's special or anything, where we really see how all of these little concepts that we talked about mass action, about movement of metabolic pathways one way or the other, how you create new molecules and utilize new molecules, all of that comes into play in the context of groups of cells. Okay? And so again, like yesterday, I'm going to post a bunch of questions. Hopefully you guys will keep asking questions and we'll continue along the same line. So here it is. Okay. Okay. So we'll again reiterate the same thing that I think both Jonathan and I have repeated now multiple times. We have ATP as our energy currency, but essentially what we're using as a means of energy is the free energy released when a phosphate group is transferred from a higher energy phosphate to a lower energy phosphate molecule. So this is again that recap of the hierarchies of what we have in cells. ATP is right here. It's about minus 30 kilojoules per mole. When you break it down, glucose and glycerol are all lower. So you can transfer a phosphate from here to here. If you have to transfer a phosphate to make ATP, you have phosphate-rich molecules that are higher on the energy landscape, like 1,3-bisphosphoglycerate. That's the GAP-TH step in glycolysis. A phosphoenol pyruvate, which is probably the most energy-rich currency we have in cells, phosphocreatine, and so on. So what you'll also appreciate from here is that you can see why you can't go back from ATP to PEP, for example. They're in the wrong direction in terms of the energy landscape. Okay? Okay, so that's summary recap one. And if there are any doubts on this, I think there aren't, but if there are any more, we could discuss this. So everyone's clear on this? Okay, great. Now, this slide changer is not working. The slide changer is not working. It was, they did something. Yeah, okay, now it's working. Okay, great. And the next part is the collective net energetics in a cell. So Jonathan ended with the whole business of electron transfer and that as a powerful way to drive proton motive forces in order to couple it to reactions to generate energy. But collectively, this is the entire summary of energetics in glucose oxidation. A lot of times we'll only go up to pyruvate, and from pyruvate, we have to balance the redox. So we just learned that in order to balance the redox, if you stop there, you have to do something to the pyruvate. You either make it lactate or ethanol, depending on the cell type you have. And the reason you do that is you regenerate reducing equivalents. So we didn't go through too much of what you do with NAD and NADH and NADP and NADPH. But what they're doing is constantly enabling oxidation and reduction so that you can move these reactions forward. So this is the big back-end player in all energy metabolism. The front end is glucose being oxidized to CO2 and ATP. The back end is all this redox balance. And then additionally, balance of things like phosphate molecules. So you have cofactors and essential substrates that we don't usually account for, but are actually very important. So very quick summary of what you get, and you don't really have to remember this, but just remember the flow. Of course, when you go in the beginning of glycolysis, you waste ATP in order to keep the glucose in there. Then you waste another ATP to make this symmetric 2-phosphate molecule. We talked about that yesterday and how it's important to make this molecule in order to split it so that you can double your payoff. This step, the back end is this NADH that's generated. This reaction has to happen in order to put that negatively charged inorganic phosphate onto the 3-carbon molecule that you create. So you create BPG. After that, you start getting your payoff steps because you put this phosphate. Now you can take that phosphate and put it on ATP, or later on, so the BPG step, you put that on ATP, you now are net neutral for ATP, no loss, no gain. And after this, you start getting your payoff, which is your PEP to pyruvate. You get your 2 ATPs extra for your energetics. In addition, if you end at pyruvate, pyruvate has multiple fates. If you stop right there, you have to make it lactate or ethanol, again for this regeneration of redox molecules. If not, you will continue into respiration. You will convert pyruvate to acetyl-CoA. You start now generating all these reducing equivalents as you are carrying out the TCA cycle. Now, the reason for all of this is coupled to the electron transport. So if you have a free electron, you need a proton to balance this out, right? And that's what this allows you to do. So the TCA cycle, or the respiratory cycle, is a combination of all these tricarboxylic acids being made and the electrons dancing on the electron transport chain. You need to have both together. If you don't have this, you'll have a redox imbalance. You'll have electron leak, you'll have oxidative stress, you won't have ATP, okay? And so end of all of this, from glycolysis, you'll get 2 NADH molecules. You'll get 3 per NADH here, and then 2 FADH molecules. Now you'll see, just like ATP and GTP, you have NADH and FADH2. They practically do very similar things. There's no particularly unique reason to have one versus the other, but biology has co-opted one and has stuck with one forever. Okay? So I think there was some earlier question on why couldn't, or would it be any different if you use GTP? It won't. Energetically, it's very, very similar. I mean, it's marginally different, but it's just marginal. Just like that, you could also use FADH2 instead of NADH, and the net effect is similar, or NADPH instead of NADH. Roughly similar in terms of redox capabilities, but biology is locked onto one. And once something's working well and it's doing the job, there's no real reason to change it from that to something else, right? So this is your whole respiratory cycle. Okay. Also, because somebody asked about phosphates, you might find this interesting. This is something we had written on exactly the flow of inorganic phosphate in cells and how that's coupled to central carbon metabolism. So if any of you are interested, I just threw this in just now because there were a couple of questions on the balance of phosphates. So essentially you have phosphates free inorganic. You also have a lot of phosphates that are bound to different molecules. You have them as polyphosphates, as pyrophosphates. You have phospholipids, which are a huge component of the cell and have phosphate molecules in them. You have metabolic sugars with phosphates on it. All of these phosphates are also cycling between the inorganic pool and the bound form into an organic form. And that phosphate is also coupled to these reactions like the GAP-DH step in glycolysis. So this constant cycling happens while you're making ATP, and cells try to maintain a fairly high concentration of inorganic phosphate at any given point of time. So the idea behind this is you have constantly a pool of cycles that are happening continuously, sources that are producing phosphates, sinks that are taking up phosphates. You maintain the overall balance. Also, because phosphates are highly charged, if you change the charge environment in the cell, you would dramatically alter what the cell is capable of doing. Okay, so
I have a question from the first slide. So when you say that biology has chosen and fixed. So how conserved is the whole cycle across biology, or at
Yeah,
least in
so
eukaryotes?
so the metabolic cycles are very, very old. I mean, essentially to have life, you needed these first. Without this, you wouldn't have all the fun other things like transcription or replication. So presumably this happened a very, very long time ago, and it stayed because it works. So we'll get into a little bit of that in just a little bit on the
Right.
chalkboard.
But do you, for example, see in some organisms that the symmetric step is skipped or
No,
something? No,
no.
it's
So
all
so glycol so the of all metabolism, the things that are really, really well conserved, of course, the tricarboxylic acid and respiration, glycolysis, gluconeogenesis. Okay, these three largely are very well conserved. What are different are how the enzymes are regulated. When the enzymes are regulated differently, you can alter the rates, you can alter the total amount, you can do that. But the actual mechanism, the chemical mechanism, has not changed in any organism that we do know of as of now. Of course, somebody will find one tomorrow, and then that's that. But there are organisms which have lost some pathways. Many of them tend to be obligate parasites or present in hosts. So they will lose a part of their respiratory cycle, or they will lose a part of glycolysis, but that's because they're relying on their host for their energetic needs. But whenever it's there, it's there. Okay. Okay, good. So
I had a question. So, like in that slide where you were giving us a summary of the
the
protein,
phosphates,
yeah, about the yeah,
energy transfer.
this. So it's like on the last three steps, the NADH from pyruvate and stuff. We'll see like +6, +4, +18 ATP.
Yeah.
So like
So this is the approximate payoff you get from respiration. From glycolysis, we get a total net of two used, two gained. In TCA cycle, with each of these couple steps, so for example, when you carry out this reaction, here we're counting the two GTP as ATP here. Actually not ATP, but it's GTP that can be used just like ATP would. These are all ATP molecules. Each of these is with this NADH that goes to the electron transfer chain and couples with the electron transfer and collectively build up the electrons and carry out um, um, the ATPase activity. Okay, so these are all coupled reactions. And this one is a cycle. So glycolysis, you see, is always drawn as this straight line. And then you draw a circle. This is your metabolism, right? this is this is a cycle because you're continuously generating these reducing equivalents, which are all going to the ETC and are being reconverted. So you the NADH. Too close. Okay. I will write this side. This same circle will work this way. There are ETC here. NADH, NAD plus, which will allow the E minus to go. Okay, and then FADH2, NAD plus, E minus, and so on. So these are continuously happening. As the cycle is going, this is being made, this is being regenerated, so this is a constant cycle. This NAD comes back here, gets regenerated to NADH, comes back here. So this is going in a circle. It's continuously happening. So any of you know how much ATP is made with one turn of the ATPase? So this is all leading to this. And finally, there's the ATPase here, which is a motor, and that turns, and that allows, so you've created an electron imbalance here. You have this rush of electrons to balance the charge, and then the motor turns. And when that motor turns, it makes ATP. Usually with one turn, you'll get 3 ATPs. So this is happening constantly at the time scale of seconds, right? And this is all going back, and this is continuing to cycle. And here you're getting pyruvate, which is feeding into the cycle to drive the cycle. Okay? And if you have pyruvate here, it goes to ethanol or lactate to balance it if you stop over here. So there are many organisms which don't rely on this too much. But in every organism that is free living, you'll still have a little bit of this because there's a second thing going on. Many of the molecules that are made here have other fates as well. And this is the other thing about metabolism. You don't just stop over here. So for example, you'll make alpha ketoglutarate over here. A lot of it is going into this cycle, but a whole bunch of it is also going away to become amino acids. So alpha ketoglutarate after a transamination becomes glutamate, which becomes glutamine. And those serve as the precursors for all your other 18 amino acids, right? Which means this whole network has to be balanced. Okay, so we'll come back to two parts. Yeah.
So it is a movement of electron across the motor that.
No, the electrons are outside in the
Yeah.
electron transfer chain that build up this negative gradient outside, and then you need a rush of protons on the other side coming in that drives the motor.
So both FADH2 and NADH would be responsible for the same amount of charge difference, right?
That's right. That's
so
right.
why do we get 2 ATP in FADH2 and 3 in NADH?
H2 and H.
Sorry?
Oh, you mean no, there are the multiple steps. Like here, so for example, alpha Kg becomes succinate.
No. My question is, so FADH2 will give a imbalance of 1 electron.
Yeah,
NADH would also give an imbalance of 1 electron.
yeah.
So the imbalance is same. So the gradient for H plus
Yeah.
is same.
Yeah.
So
No, they're all
the
going
turn-
together. They're all going together. It's not like they're separated.
no, I'm saying it's just a physical change, right? The
Yeah.
change in the gradient of electrons,
Yeah,
the charge.
yeah.
So shouldn't both the molecule be responsible for the same amount of turns?
No, the turns are not happening. The turns are in the final, in the ATPase, the final step, which is separated from the electron transport chain. Okay, so there are 4 complexes, complex 1, 2, 3, 4, and then 5 is the ATPase motor, separate one. So here, this is building up the electron gradient, and this one is just the motor, which is being driven by the gradient that's built. So it's a cumulative effect of all of it. It's not separated at the level of these. There are 4 complexes, and this couples to the 5th one, which is the motor. Yeah? Okay, so what we're going to now talk about in about 15 or 20 minutes is this. What you have already, I think, appreciated is that there are reactions which are mutually incompatible. If you're having one reaction, you won't have the opposite reaction, right? Any reaction goes from A to B or B to A. And of course, we're saying many of these are enabled by enzymes, right? And it's usually driven by the collective net free energy of the entire pathway. The collective free energy will be negative, so you go from higher to lower. But now when you have one reaction going a certain way, it's highly unlikely that it'll go back the other way, because now you have these multiple rules going on. One is that of mass action. If you have a lot of this, you're going to end up with the other one and less of this in the other direction. The second is this delta G barrier, right? Supposing you're going this way and it's net negative here, it's going to be net positive the other way, which is kind of hard to do. So, but we do know that these can happen in cells in the opposite direction. So how do cells deal with opposite reactions? Well, that's really the essence of what you think of when you think of anabolism and catabolism, which was introduced earlier. One is the breakdown. So glucose oxidation is considered catabolism. You're breaking down glucose to release energy, and build up of something is anabolism. Usually those are energy consuming processes in terms of net energy. So catabolic processes, net will release energy, and anabolic processes, net will utilize a lot of energy.
Metabolic cycles that you drew there,
TCA cycle, yeah.
they are happening in the cytoplasm, is that right?
This is depends on the organism. In eukaryotes, this happens in the mitochondria. In bacteria, they actually happen on the membrane. It's different, but they don't have the mitochondria is a bacteria.
Sure.
And in plants, you also have chloroplasts where you have electron transfer.
But how does the input to this metabolic cycle?
Yeah, very good, very good. So these are all controlled, right? So I've drawn this nicely, but actually what's happened is this is cyto and this is mito. We have pyruvate over here. Pyruvate has to be taken in through a very, very important protein called pyruvate dehydrogenase. If pyruvate dehydrogenase is there, not there, you can't utilize pyruvate, take it into the mitochondria and make it into acetyl. So at all of these, whenever you have compartmentalization, you have a gatekeeper protein to do something over there. This is universal. Of course, if there is no compartmentalization, then this problem will not be there. Now, this is also true for other things. So for example, the NAD, NADH that's made inside the mitochondria is a different pool from the NAD, NADH that's in the cytosol, which means cytosolic redox balance is controlled by this pool in the cytosol, and the mitochondrial redox balance is controlled by this pool in the mitochondria. They're not freely exchangeable. A small amount of it can be taken in and out by transport.
Maybe a follow. So when you said this electron transfer, that must be happening in a very coherent way. So therefore, the actual spatial volume in which this is happening.
Yeah.
I mean, if you just let them freely flow, they'll just diffuse around. You'll not get any coherence around that.
You know, actually, so Mitchell's chemiosmosis is now what, 50 years old or something? He proposed all of this happening. And actually, we are at about the same place as his original proposal over there. Exactly how everything is contained and kept over there, we are still hand waving. So I will also hand wave. But it has to be that way. I mean, that is the essence of the whole chemiosmosis theory. It's all controlled, regulated. You'll have electrons building up, creating a protomosipose, motors working in perfect synchrony with that. It happens. It happens here. It happens in plants, in the carboxysome, and so on and so forth. So it's there. Okay. So I'm going to use that side of the board now. So essentially, we could understand a lot of this making and breaking, taking our favorite pathway, glycolysis, favorite only because it's kind of the most important, and the opposite of that, which is gluconeogenesis, right? So gluconeogenesis essentially is the making of glucose from smaller precursors. So which one is catabolic, which one is anabolic? Just to wake you up. Okay. Yeah. So glucose breakdown, glucose oxidation is catabolic. Gluconeogenesis is anabolic, which means net should you be using or gaining energy from gluconeogenesis? You have to be using energy, right? We went through glycolysis and you saw how much energy is made. You're going in reverse, you have to use that energy. So why do cells do it? Why do organisms do it? Storage of what? Yeah, but whatever is there is just going to be broken down, no? When there's depletion of glucose, you'll use a store. So why do we do this? In part, yes. You allow other energy sources to be converted to glucose. Why would that be important? That's on the right track, yeah.
Because not all cells can metabolize all of it.
Yeah, so that's what we see. But if we ask why, so it's right, you're right. But that's what we observe. But why do we need it now?
Can only enter the TCA cycle. Can't have glycolysis for all. For example, fats and proteins, if they have to enter the TCA cycle, they all have to be converted.
Okay.
That's why convert everything.
Okay. Good. So now let's just work through gluconeogenesis again from first principles, but this time without even the structures. I'm just going to draw circles on the board. Okay. And we'll see how this holds. So what's glycolysis? You start with glucose. right? You're just putting one phosphate over here, right? Then you're going to make this a symmetric phosphate here. So to do this, you need ATP. To do this, you need ATP, right? This is your fructose 1,6 bisphosphate and this highly regulated step. The reason we do this is now superb to make you these 3-carbon G3P and so on. Then you have 3-phosphate and making BPG. This is your GAPDH. I'm just going to mark a few key steps. Yeah. So now here we start getting payoff. You start getting payoff over here, right? And then you get PEP, finally pyruvate. So again you get ATP, right? This is glycolysis. Net gain two ATPs at the end of all of this, because this is 2x, 6 has become 3. Great. How do we go backwards now? What would be the challenging steps? So, is gluconeogenesis the reverse of glycolysis? That's the first question. Endpoint is the reverse of glycolysis. You have to get to either G6P, or you actually get to only G6P. You don't get to glucose. Why? Because we don't want to release glucose outside. So we stop at G6P. So gluconeogenesis goes back to G6P, right? Just, yes, I'm sorry, that's mine. Just kill it. Whatever which, thanks, yeah. Thanks, thank you. Sorry about that. Okay, so back to glycolysis, more interesting than that phone call. What would be the challenge in going backwards? Just look at this and say which steps are going to be the most difficult to do. Yeah, so we'll start backwards. We're going backwards, we're starting with pyruvate. Would this step happen? What did we learn about PEP? It's right high on the energy scale, right? The highest on the energy scale. Actually, there's no way to use ATP to put a phosphate on pyruvate to make it PEP, because the energy barrier is too large. So what do cells have to do to start gluconeogenesis? They have to find an alternate way. And that's the point of metabolism, finding many alternate ways to get to a destination. Right? So the clearest illustration of the need for this is gluconeogenesis. Now cells do a lot of this. We have good reason for doing gluconeogenesis. There are actually two reasons. One is, if you don't have glucose, you have an alternate carbon source. How do you start utilizing it effectively? The only effective way to generate all of this is through glycolysis, which means you have to find a way to get to glycolysis first. And the way you do it is to make glucose. You could start with anything. You could start with pyruvate, you could start with lactic acid, you could start with ethanol, you could start with lipids. Lipids will have to, again, go through a little bit of beta oxidation. We're not going to go into all of that. They'll break down, and then you'll still start making molecules like pyruvate acetate. You'll make acetate, which can convert to pyruvate, right? You're starting here. It's very easy to get here to pyruvate. But then you need to find an alternate way to do this. And so this step, this is why this is such a bottleneck step in this whole process. You can't go to PEP. You go around, you work your way around. You will convert pyruvate to something else, typically to oxaloacetic acid. And then you'll utilize lots of ATPs. You'll utilize multiple molecules of ATPs in pyruvate carboxylation, actually. You'll go all the way around and work your way backwards. So pyruvate converts to oxaloacetate. Oxaloacetate converts utilizing ATP to PEP, and it goes a long way around. So that's one way where you get over this huge energetic barrier. use an alternate pathway. This is why we have alternate pathways, yeah. You can use the mic. There's a mic somewhere.
So you said if there's an alternate carbon source and we want the entire glycolysis and TCA to happen, the first that carbon source has to be converted to glucose, and that has to happen.
Yeah. So just to clarify before that, just before your question goes on, why does it have to go all the way back to glucose?
That's my question.
That's your question. Okay, good. So why does it have to go all the way back? Why can't it, for example, just stop here? Right? This is kind of more useful. You make it all the way here and then you break it down to get energy payoff. Good. So why? You mean just sitting around, right? Like these intermediates won't just sit around. So let's put it this way. What do you get here? You get 6 breaking to 2 and doubling, right? If you just stay at 3, you don't get that net gain. What happens here, for example, is from pyruvate to go to PEP, you're using 2 ATPs. You'll get only 1 ATP back. So it's kind of, the rest of the energy is dissipated, it's gone, everything. So you don't get a payoff. You have to work all the way back. So it's highly inefficient in that sense. It's very costly to the cell. You're using a lot of ATP. Actually, for gluconeogenesis, you end up utilizing 6 molecules of ATP to make 1 molecule of glucose. It's all net loss. But you're doing it so that A, later you can use it for glycolysis, the payoff is very good. And B, you actually have lots of uses for many of these other intermediates. So I've drawn this linearly. There are many other things that branch off glycolysis. One of them, very important, is our ability to make ribose sugars and nucleotides. That's called the pentose phosphate pathway. Without that, we won't have the precursors with which we can make nucleotides. And without nucleotides, we're not making RNA and DNA. So you have that. You also have branches off here at the 3 carbon step to make certain types of lipids. You also have intermediates that come out over here that go into amino acid metabolism. So you need to keep working backwards if you don't have ready glucose also. Okay? Okay, good. So one key step here in gluconeogenesis, if you have to go backwards, you don't go back this way, you go back this way. Most of this is actually pretty straightforward. It will go backwards. You'll get to here, and then you'll even get to here. Now, is this step easily reversible? It actually is. So what did I say yesterday? There's this, you put a phosphate, you add another phosphate here in order to break it for the payoff. But it's not that hard to remove a phosphate from here to go back here. So there's a different enzyme which does this, which will go backwards. Okay, so you can make fructose 6-phosphate. It's highly regulated, but this is highly favoring the forward direction because you're using ATP to put this on. There's another enzyme that is highly favored in the reverse direction to enable you to get here. So the 2 key differences in this whole gluconeogenesis is first, this big part, this is the hardest part. Once you solve this, the biggest energetic barrier is kind of minimized. And then the other one is this. This is the most highly regulated step for glycolysis. So if you want to go the other way, you have a different enzyme to do it. And that's the highlight of alternate pathways to get to a destination. Okay, any questions over here? I can't hear you. Can somebody pass a mic?
So these different enzymes, one for forward and one for backward, are these in different cells so that these processes are regulated in different cells?
Very good question. I will actually talk about exactly that from one of our studies. But in general, every cell, the genome encodes for everything. So the relative expression of what you have is in part going to determine which way your reactions are going. But also, the amount of the substrates present will determine which way the reaction is going. So if you have a lot of glucose, everything is geared in favor of breaking down glucose. You will only break down glucose. You won't have much gluconeogenesis when you have high glucose. Because mass action is built that way. You can't fight mass action, you can't fight laws of thermodynamics. The only time you'll start going the other way is when you have different starvation. You don't have enough glucose, but you have an abundance of other early precursors that lead to pyruvate. Yeah? Okay, good. So now, if I have to ask you a slightly philosophical question, origin of biochemical pathways, which one must have come first, glycolysis or gluconeogenesis? Why do you say that? It is a big gain trade-off, but would it have come first? What did we have in an early universe? Yeah, origin of pathways, right? When we're starting with life, what would we have had? At least according to the, current hypothesis ideas, Urey-Miller experiments. In the Urey-Miller experiment where they just heated a bunch of carbon and nitrogen, they didn't get glucose. You get amino acids, you can get a carboxylic acid. So it's very likely that, this is all speculation, but it's fun. But it's very likely that gluconeogenesis had to have evolved first. What do you need for that? Some way to get to pyruvate, right? And so let's say you don't have pyruvate. The easiest way you would get to pyruvate is from some amino acid. Let's say you have, so pyruvate is this, right? This is pyruvic acid. Easiest way to get here would be, say, from alanine, right? So you have what do you need to do? It's pretty straightforward. You just need to deaminate this. How do you deaminate this? You need some recipient. You have another carboxylic acid, alpha ketoglutarate. You just have to transfer this amine to alpha kg, and you get this, and you get this. This amine you can transfer to this, and you'll get glutamic acid, which means amino acids and carboxylic acids must have had to be there for a way for you to get pyruvate. Once you get to pyruvate, you have to do all these challenging steps. And even for this, you have another set of acceptors to transfer things around. So you'll convert pyruvate to oxaloacetate to PEP. You slowly work yourself backwards. It's amazing. All of this must have happened at some point of time, billions of years ago. But it did. So presumably, gluconeogenesis came first. But there's a problem. What's the problem? So definitely glycolysis has to have come after gluconeogenesis came, and then we co-opted to the system. Because the gain, the benefit is so immense once you have glucose, right? Breaking it down is gold. But how do you make glucose in the first place? All of this part is easy. You can do this, but you need energy to do this. So there's no, you need to have ATP or something there already, which presumably means, and since the carboxylic acids are there, presumably I think electron transfer need to have somehow come before gluconeogenesis could happen, because you first need an energy source that you could transfer. So anyway, this is just speculative. It's fun. We have no idea, but if you have to logically break it down, this is how you would do it. Okay, makes sense. It made sense to me. Okay, let's move on now to slightly more research part of it. Okay, so now I'll try. Yeah. No, good.
Do they have the?
They have everything. So everything that we have now has everything, right? You have glycolysis, you have gluconeogenesis, you have the TCA cycle, you have electron transport. The only ones that don't have it are those where if you reconstruct it, you know that they have lost it later on. You get it? So the question is, do we have archaea or something, examples which don't have this or have lost this? Everything, even low-key archaea and all of them have everything. And those that don't have lost it much later in evolution as they became obligate parasites or something like that. And you can draw these trees and construct it backwards. We don't have an example without it, so we can speculate. That's it.
Sunil, so I have a question. I don't want to hijack the the questions for the students. But so what about these papers in which they, I think from Crick Institute, I forgot the name of the group where they managed to what?
Marcus Ralsa?
Yeah, exactly. Right, where they do glycolysis, I
Yeah.
think, without energy source,
Yeah.
just by fine tuning, as I
Yeah.
understand, the ionic concentrations.
Yeah.
and-
Yeah. So what?
Can something like this also happen for glucogenesis? I would
Yeah. So
imagine?
the idea, what Marcus's group tried to do is reconstruct an artificial way to do glycolysis, where they artificially put in or imbalance the proton electron ratios and try to drive redox reactions. But the whole point of that is you have to have a way to drive the redox reactions. And so you do need something like NAD, NADH. Otherwise, you can't go backwards. And energetically, it was.
I mean, you need to tune the ionic.
Yes, yes. So they got very, very, very inefficient something happening.
It was very inefficient.
It was extremely, extremely inefficient. But they're
non-zero,
trying to get at
non-zero.
this kind of a question exactly. This is
Okay.
like to see what are the limits of possibility and what needed to might have happened.
Okay.
Exactly that, yeah. Yeah. Okay. So we'll move on now to a little bit of now putting all of this in context. By the way, separately, so now you also appreciate how important the whole network is, right? So what we do, what we can do today is model some of this at the level of a network. Nitish, who's a TA, will maybe go over how you could build a genome scale metabolic model. I'm not going to get into too many details of that, but essentially what you do there now is convert a lot of this entire network of reactions into mathematical equations. And the way you do it is make three big assumptions. One is, of course, we talked about steady states, right? And non-steady states, and also order of reactions. When you do a genome scale metabolic network model, you first assume that all reactions are at zero order kinetics. Okay? They're all operating at steady state. Now, this is not really true in a cell because things are constantly changing. But you make that assumption to start getting some useful predictions. So basically, it says all reactions that are present in the network are operating at zero order kinetics, which means it only depends on how much of the enzyme is there. And then what you can do is take, based on the genome, annotate or classify what reactions are present or absent in that genome or in that organism. Then you put essentially presence of a pathway enzyme, that's a check. So you have a score or a matrix which you build with one for the enzyme and one for the metabolite. And you put a yes, yes, or no, yes, and so on, and build a matrix. And then you build the whole network, and then you can run the whole reaction to say this is what will happen. Now, on top of that, you can put some scaling parameters for number of molecules of each of these present. You usually don't mess with the metabolite. You just say, yes, it's there. If it's there, it's there. But what you can do is change the amounts of the enzyme, which will scale how active or inactive a particular node in a pathway is. So it's an assumption. This has no kinetic information. It has no allosteric information, which means about 50% of regulation is lost. But it's still very useful because what it does tell you is what happens if you dramatically change any one node in a network. This completely relies only on mass transfer and nothing else. Okay? So we don't have really time to go into all of this because if you go into this, we'll miss out on all the fun, what you can do with metabolism part. But maybe Nitish will go over that in at least how to build a genome scale model in tutorials sometime there. Okay, so we'll move on now to some examples where we learned a lot about some of these kind of things as we found some phenomenon. Essentially, what you want to do with all this metabolism is contextualize it to cells and then cells to tissues and so on, right? And we know that there is this enormous diversity of cells. Even in the simplest organism, you have enormous heterogeneity in their cell states. And always, and this is again something Jonathan alluded to earlier, the way you think of cell states, this is a parallel world where they haven't dealt with metabolism and so on and so forth for a long time. And so the idea there is cells are traveling down this Waddington landscape, and then they would fall into these different valleys where they would remain trapped in that state, and that's how you get differentiation. But essentially what we do know extensively is that cells are a function of their environment, and the environment essentially fundamentally controls metabolism, right? So I'm just asking you to keep all of this in mind, and as we go along, we will try to see how this space and this space will intersect potentially at metabolism. So I'm going to now talk about this idea of heterogeneous cells, which we see very, very widely. Every single experimental biologist encounters this, and we just call it heterogeneity. You will see this in a cancer tissue, you will see this in bacteria, you will see this in protists, you will have transitions of cells where you have, so for example, this is a dictostelium, it's a type of solitary amoeba. When you starve it, it will become a complete multicellular aggregate of cells. And this is reversible. You can also see that this heterogeneity can happen, break, form, break. There's a lot of continuous dynamic processes going on. Okay? Essentially, if you take simplest unicellular protist or a bacteria or a yeast or something like that, and you find ways to bring them together, you can start getting aggregates that are working together to do different things. So that's really your multicellularity, whether it's a plant or an animal or something much simpler. The simplest multicellular animal is this, again, it's an amoeba, Tetrabena socialis, which is a multicellular organism of 4 cells. And it is an obligate multicellular organism of 4 cells. You have wonderful examples like these flatworms, planaria, for example. This is the adult normal planaria. You could chop this up into how many other pieces as you want, and they will always regenerate to give you the same animal again. Doesn't matter how you cut it, you slice it vertically, horizontally, it'll regenerate. But essentially what you have as a requirement for a collection of cells are these following criteria. I think this is a very nice way to think about it instead of thinking of animals or tissues or something like that. This is a very nice paper by Nicholas in 2014. You first have to have some kind of orientation or polarization and adhesion. Adhesion, in the way we think of it in multicellular organisms, is irreversible addition. But all these examples and the dictostelium, for example, tell you that this irreversible addition is an unnecessary overcomplication. What you do need is the cells to be together in some kind of a stable way. Yeah? And then you start having differentiation, allowing exchange of material between the cells or the tissues, and eventually polarization. And that's the simplest, most kind of uh, parsimonious explanation you could have, or a definition you could have, of multicellular. But essentially, we'll come back to today's talk. What we're actually looking at is some form of phenotypic heterogeneity. Now, one of this is the real Donald Trump, the rest of them are fakes. But as far as we're all concerned, I think of them as Donald Trump, right? They're all the same. But there's another way to look at it. If you have a group of cells, they're all slightly different, but they're all essentially doing the same thing. They're one cell type. The other is your truly differentiated groups, like your stormtroopers and rebel fighters. They are distinct cell types. And what's really important is knowing what are the differences between these as opposed to the differences between these. So the clones are not exactly, stormtroopers are not exactly clones. I mean, they look the same from outside, but they have differences within. And sometimes, especially today, you'll see a lot of noise, literally noise, about differentiating between these. It may not amount to much. Okay, so keep that, just keep that as my opinion, and we'll move on. Okay. So the other idea that comes from having groups of cells, yeah, there's a question back there.
Before you move on with the fun example. So in the previous slide, the socialist, that one, so all the cells are coming from a single cell, which divides into
No,
4
they're not. So in a social amoeba, you have individual amoeba living in like a outside a pond or something. During starvation, they come together and aggregate. Now they're all the same species, and at least until recently, it was thought that they were clonal, because typically in an area you'll find just a clonal population that's expanded. But there are examples of non-clonal cells coming together to form an aggregate, stable aggregate that moves. It's a slime mold, then it starts moving, it starts foraging for food and doing things that are far more complex than a single cell can do.
So the 4 cells, that particular one, right?
Yeah.
How do they count? Is there a variation to that? Like, is it like
That's a nice
sometimes
question.
3, 4, 5, or is it always 4?
No, that's always 4. That's the Tetravena socialis. That's always 4.
So how
I
do
don't
they?
know. I don't know how that happens. That's very cool though. I mean, there must be other interesting things going on. Why have 4? I mean, why stop here? Why even have 4? I don't know. Good question. There must be a counting mechanism. There must be a limit to sizing, whatever, scaling laws, sizing laws. I don't have an answer to it. Okay. Shall I move on? I have no idea, so I don't want to speculate. Okay. So the other idea I'm going to talk about today is this idea of not just heterogeneity, but being organized in heterogeneity. All of these and all of these do distinct tasks. And that's your idea of division of labor. What you have in division of labor is a group. I mean, in economic terms, you have different workers doing specialized tasks, doing one job, doing it very well, so that collectively you can do a lot together. But you see this a lot in cells, even in simple microbial cells, whether they're clonal or not, where they live and work together to carry out specialized tasks. So sometimes they do functional complementarity, opposite tasks that one can do and the other one cannot. Sometimes they just have synergistic advantages for the whole community to grow better than the others. And you also have various other forms of positive assortment and so on. But all of these define your division of labor. We're going to look at both of these ideas in the context of groups of cells, but sticking to this old idea of what glycolysis is important. Okay. So we come back to this. Basically, the question we started asking some time back is how do you get organized phenotypic heterogeneity? Okay, and the idea we had was in every single example we look at, there seemed to be a strong trigger that came from some form of starvation or nutrient stress. If you read the literature, they just call it nutrient stress. It doesn't try to define a logic to it. Say starvation. Starvation causes cells to specialize. Starvation causes cells to differentiate. We thought, okay, taking that well-established knowledge, can we start looking at something to see if we can find very simple ways of organizing heterogeneous cells together. And this again comes back to this single most important idea in regulation in biology. Timescales are everything. And you eventually look at final form and final function, but these are the timescales that we operate at, right? And we talked quite extensively about this in the morning's lecture. And what typically changes the fastest or can change the fastest is this, but it is not in isolation. They're all in crosstalk with each other continuously. So a metabolic change will signal to something which will affect proteostasis and transcription, which will eventually affect cell fates. But as you change, your state changes. Once you have a transcriptional program, the entire state of the cell changes. So this feeds back and changes this. And so it's a two-way street. It is not a one-way street in any which way. And this is important. It's dynamic, it's fast. They're operating at time scales like this, but they're working back and forth with each other. And collectively, you could then amplify signals to end up with eventual stable outcomes. At least that's one way to think about it. Okay? Okay. And the end point of all of this is you're changing your overall resource allocations within cells, but also between cells. As you have collectives of larger numbers of cells, your resource allocations towards what would be a growth program versus any kind of an alternate program would change based on the changes in your metabolic allocations, which come from this constant crosstalk between each of these. So you can actually study this problem at any of these scales, and you will be answering questions, as long as you are aware that you have to go back and forth between these scales. Yeah? Okay, very good. So what we started to ask was this idea of heterogeneity. And typically when you look at cell states or cell fates, the classical way is to take a gene-centric approach. There's this gene required for differentiation, or there is this developmental milestone that comes from a gene. If you now break it down to like a first principles kind of a question, you're actually asking the same question differently. You're asking what kind of molecules are needed to bring cells together. What kind of molecules are required for a certain type of cell to sustain itself? So can you have then groups of cells making different types of molecules, and how do you go about getting to that point, right? So it's really a biochemical problem. And so to address this, we wanted a very, very, very simple system. We didn't want all the complexity of a tissue or an embryo developing or something like that. Because the question was very simple. If we get just a couple of different states, how would we look at it? So we chose a very nice, very, very well established model organism. Again, something that was introduced in the morning, the use of different model systems. We use yeast, mostly because yeast are the most important organism in the universe. This is why we all exist, or something like that. There's actually now I can say this because there's a Nature paper which says that human development happened because of yeast fermentation. So if it's in Nature, it's established. Okay, so the model is yeast. Now, typically, yeast have been used as because they are unicellular eukaryotes. That means the whole point of it is each cell is doing its own thing, each cell behaves its own way, and we can get to molecular mechanism. So that's how we found the cell cycle, for example, or growth laws in single cell organisms and so on. That came because it's so easy to use and manipulate. But this is something that actually is very well known to everyone except the lab researchers of yeast. In the lab, when you grow them in rich nutrient conditions, we all grow things in rich nutrient conditions in the lab, you form a colony like this, right? Except that all yeast in the wild don't grow on unlimited glucose constantly. They grow on much more limited resources. And in those conditions, especially when you have lower glucose, you form colonies like this. Now, of course, everybody knew this, and for us, it was just a little side exploration because this looks nice and you see morphology, some orientation, architecture. We thought we would just try to break this down more specifically to see what we could learn from this. This is work over a period of years, okay? And so this first story was published, and we'll stop with this one. And then next week, we'll go into something, next class, we'll go into something more detailed. But so what do you see here? You see this is very different from this, but you see very nice organization. And what we observed, what Sriram, who was the postdoc working on this, observed, is that this is just under a light, bright field lens. So it's not high resolution. These colonies are huge. So these are about half a centimeter in size. They're quite big flat colonies. But if you look under the microscope, you see this beautiful patterning inside them. Okay, and it's very, very reproducible, very robust. Every time the colony develops and matures, it looks like this. We had two regions, one was light and one was dark. So we just called it light and dark at that time. And that name has kind of stuck. So, but it's nothing special about dark and light, it's just optical density. There are more cells here and they're slightly different. So they show up as dark because this is a bright light source that's shining on the colony. Okay. But we just started looking at this and asked what these cells were all about. And we did this keeping in mind this basic idea of what is required for cells to grow in very low glucose conditions. So what did we learn? In low glucose, the process that you need is gluconeogenesis. There is no way around it. You can't get around it when you have low glucose. These have everything else, they just have low glucose. They have amino acids, they have all of it. So the assumption is all cells should be doing gluconeogenesis at a fairly high efficiency, right? This is a necessary process. We started to look at that and just tried to find the distribution of gluconeogenesis across the cells. There are different ways to do this. I'll just talk about a few experiments along the way just so that you know how you would do an experiment to test out something like this. What we did was build fluorescent reporters that would indicate any one state. We basically had a red reporter for gluconeogenesis. For that, we were using an enzyme, pyruvate carboxykinase, which is over here, this reverse step, coupled to a fluorescent RFP, a red fluorescent protein, right? And so the assumption is you'll have gluconeogenesis everywhere, but this is what we saw. All these dark cells were very, very highly gluconeogenic. All these other cells over here had very low gluconeogenic activity. So we see beautiful spatial separation of a metabolic process. And this is the process that is supposed to be there in all cells because the condition has next to no glucose. So this was nice. And so what were these other cells, these light cells, all of these over here, especially at this rim? What kind of state were they in? And so we thought of possibilities and we looked at different reporters, but essentially what we found is that especially these cells, all of these over here, were highly glycolytic. So same idea, we use some reporters, and these reporters are, in this case, for the PPP, but that's also a reflection of high glycolysis, very, very highly glycolytic. And to really see if they were really so, there are two ways to look at it. I said you might just have the enzymes and maybe the pathway is not active. So we tested to see if the pathway was active. And for doing this, we talked about a pulse chase earlier. We did something similar. Here we looked at stable isotope place trace of the flux of the pathway, right? What we see, in this case, we didn't do the pulse chase. The pulse chase comes next, sorry. We looked at the total amounts of intermediates of this pentose phosphate pathway. We find that these light cells had very high PPP, this one had low. So these light cells are really in this highly glycolytic type state. Okay, so we have a paradox over here. We have a collection of cells. These are millions of cells, so thousands, tens of thousands of cells. Most cells are supposed to be gluconeogenic, but we have a large fraction of cells that are highly glycolytic. And we just discussed that these two pathways are actually not supposed to happen at the same time, but clearly now they're at two different places. They're happening at two different places. But this pathway shouldn't exist at all because there's no glucose in the system, right? So how is this happening and in this organization? How do you,
In the previous slide, if you could go back to
one.
the one before as
Yeah.
well.
Not a great picture.
Before this. Yeah, so
That is
if you
great.
look at the figure here, it looks like the ones inside, like not at the boundary, the ones inside are more gluconeogenic.
Yeah, these.
And yeah, and
or
the
these dark
next,
ones.
exactly. So, and the next one shows that these cells on the outer
These,
are,
it's actually not exactly this, it's this whole section. But
right,
we see these two states. If you go back to this whole colony, we
right,
see this clear differentiation of states only around here. The core
right.
is metabolically very inactive.
Inactive. And the ones,
Both, it has very little gluconeogenesis or glycolysis. And that's
Right.
expected because they're
Yes.
probably extremely nutrient starved.
right, right. And the ones in the exterior are, the pink color shows up, right? So
Yeah.
they are more glycolytic.
Highly
Okay,
glycolytic.
so I was wondering if there is a factor of diffusion that is also playing a role
Yeah.
and not majorly on low glucose or high glucose.
Yeah, so hold this thought because, okay, so two parts to this. Of course, these were all first things the reviewers would have asked, so correct. First thing, this is the mature colony. It takes 6 days to get here. Okay, so the initial colony is uniformly gluconeogenic everywhere. Like when we start and see the day 1 and the day 2, approximately day 3, we'll have some timelines coming in later. Uniformly gluconeogenic. Second, we did a number of experiments to say that this is not a simple diffusion parameter. We'll come to that in just a minute. Hold that thought. If it's not clear, we can ask this question again. Okay, good. So we are here. Now, how do you get this kind? What you also notice is this pattern is very reliable. It's very predictable. It comes every time. This is just a little aside. How do you get patterns, complex patterns in biology? I thought I will just introduce this. It's not really relevant to today's talk, but it's a nice idea that many of you might be interested in or be aware of. Essentially, the most common form of patterns that you see, which especially developmental biologists have looked at, are what are called variations of Turing patterns. What you have are gradients of morphogens at different parts of a tissue. You could have, say, Wnt over here and something else over there. And the idea is that now at place X, there's a lot of a morphogen. At place Y, it would go down at this concentration. And based on that, the cells adjacent to it would receive different intensities of signals. And you could get a pattern like this. If it's one morphogen, you'll get a simple flag like this, the French flag. If you get two morphogens, you'll get something more complicated. You'll get the Union Jack. And if you have interactions between the morphogens, this is like the little networks that Jonathan just explained in the previous talk, a feed forward and feedback loop. You can start getting more complex oscillatory patterns, which will result in more complex patterns like this, okay? And you see this everywhere. So these are examples in biology of famous visible patterns, zebra stripes, fish color, cone snail shells, and so on. But you also see this internally during development in the formation of differentiated tissues. This is an aside. We're now coming back to the metabolic part of it. Okay. But in a way, we were thinking along these ideas. And we thought, okay, what if then the gluconeogenic cells, as they're growing and proliferating and building up, start putting out molecules that will allow the other cell to switch state in a useful kind of a way. So it has to be some kind of a resource that can be consumed by cells, because otherwise the cell cannot switch state and actually carry out glycolysis. So we built some models for this, and these are very simple models. This was done mostly by Vaibhav, who was a student with Sandeep Krishna. Now he's a postdoc doing famous things. Um, of course. Yeah, right. So what do we have in the medium? There's no glucose. There's very little glucose. There must be some from breakdown products. But we have lots of other things. We have complex polypeptides. We have amino acids. We have other micronutrients. We'll have some amount of lipid. All of that is there in that media. It's more like a natural broth of things that has a mix of things, except glucose. So it's being taken in from the environment. These are colonies that are just growing on, it's like mold on your bread, right? It's exactly that. It is mold, not on your bread.
In this
We do.
colonies?
We have all those numbers. In this case, there aren't too many dead cells. Actually, the colony has very, very few dead cells. Even at the core, these are metabolically fairly inactive, but they're not dead. If you take them out and put them in fresh media, they all start growing. The fraction of dead cells is less than 5–10%. Very small.
The low glucose media that's being used for the growth, is there a specific defined concentration of glucose that exists? Why I'm asking is because for cancer cells, the media that we use, high glucose has 25 millimolar, low glucose has 5 millimolar. But is there a defined concentration?
So in yeast, we typically grow yeast in 2% or 4% glucose. We start these colonies in 0.05% glucose. It's much lower, but it's not zero. But there is a little glucose, and you need that little bit to get them started. Otherwise, they really don't grow. But it is a lot lower. It's not zero glucose. We just reduce it to what would be low and quite acceptable. Like in the environment, you would find that amount of glucose in any place. Okay. No. Keep interrupting.
Carefully streak the cells from these two different regions. They all recover
Yeah.
completely.
Of course.
There is
So that's an obvious question. This is entirely reversible, right? You could take any cell from anywhere and seed a new colony, and in the end, you'll get exactly this colony again, regardless of where you seed it from. You literally can do that. Of course, somebody had asked for that. We did that. Yeah.
It would take a few generations for the cell to recover.
It takes
right?
many generations. So the growth time or doubling time of yeast is approximately 90 minutes. And we're seeding these colonies and we're looking at these after 6 days. So it's late, very late.
Do you see that for example, for starved cells, which are in low metabolic state, the growth rate is different?
Yes, they are slower. So they're about 40 to 50% slower. So that's actually kind of a little take home at the end, why this collective advantage comes. You have two growth rates in the whole community, and it allows the community to grow. Yeah. So there's about a 50% difference in growth rate. Okay. So we came back to how this is just an idea of how to build a very simple coarse grain model. This is not a very fine, detailed, precise model incorporating various parameters. It's what you would call a very simple coarse grain model. The only assumption we made is you have two cell types eventually. They're dark and light for simplicity. The assumption we'll make is because we know all cells initially were kind of gluconeogenic, these cells are putting out something, factor X. And if it's being put out, all cells have equal opportunity to take it up. But some cells switch to taking it up and utilizing it. Once they start utilizing it, they switch their state and they become stable utilizers. Right? So then you'll have a system that self-organizes into a system of producers and consumers. So this is the kind of the process. But what we would then do is put this on a 2D grid. You have a spatial lattice, and you can try to have decision points here. If you have cells, they'll all be putting it out. Resources are spread out. And then you could stochastically see if the cell takes it up and utilizes it. And if it has enough utilization, it can actually divide. And if it divides and stays in the same state, it stays in that same state, you could start getting patterns in expansion. And that then fits into an algorithm. Something like this, where you put decision points, how much resource you take up, how much you utilize, and whether then that's enough to actually allow you to divide, or you just remain in the same state and it's not enough to divide. So this is how you would go through building a fairly simple coarse-grained model to see this kind of differentiation. Remarkable thing is this really baby simple model gave us colonies that look like this, which is very, very similar to what we see at the real colony. And in controls, if we either blocked gluconeogenic cells from putting out a molecule, or if you forcibly said you don't get enough of the molecule to prevent switching states, you will get small non-expanding colonies that look like this. So now, really, if we come back to biochemistry and the whole business of what we were talking about today, we need to find out what the cells are doing, right? And so if you want to do this now you go back to doing old-fashioned biochemistry to identify a molecule. But we have criteria, and what the models help us do is say what is likely to be possible versus what is likely to be very unlikely. Okay, what is unlikely? Sorry. Yeah, that's what good models are really useful for. They eliminate all the improbable or least probable, least likely events. And this was the beauty of this very nice model. So we have criteria for the resource. It's available. It has to be put out in the environment. It has to come from gluconeogenesis or a related pathway. It has to be taken up. And this molecule should then be perfectly utilizable by the cells that switch, right? And so then as a control experiment, if we block either the production or the utilization of this resource, we should not be able to create this kind of a morphology. And so this is what gluconeogenesis does. We go all the way back here, but then we don't stop here. The way mass action works is when you're working backwards to making small quantities of glucose, it'll be fluxed through highly active reactions to make other molecules. And these are the kinds of molecules you'll make from gluconeogenesis. You can make glycogen, which is a famous gluconeogenic store, UDP glucose, chitin, glycosylation, and trehalose, right? Of course, we eliminated all of these through a series of experiments. We won't get into that, but really what already stood out is trehalose, because trehalose is actually a 2-molecule of glucose molecule. It's a stable disaccharide, which is essentially 2 molecules of glucose, which means if you have this and you break this down, you now get 2 molecules of glucose to start glycolysis, which is great for cells, right? What we saw is as the colony develops, the amount of trehalose builds up externally, which means it's going up. And then we could see that the cells, some cells specifically took up trehalose and broke it down. And I just want to explain how we did this experiment, because if you're starting to think of what kind of data you need to start to convince yourself, you need to do some direct experiments like this. So essentially here we've done the equivalent of a pulse chase. We measured metabolic consumption and flux. What we do is to the whole colony, provide a small trace label of labeled trehalose. In this, every molecule of carbon that's in trehalose has a +1 mass because it's a stable isotope of carbon. So the net mass gain in trehalose is a +12. And we can now track this and differentiate it from existing pools of trehalose by monitoring the mass of this versus native trehalose, right? You do that by mass spectrometry or by other methods. We use mass spectrometry. And we could very nicely see, when we provided this exogenous pulse of 13C trehalose, the dark cells don't take up this trehalose much. The light cells take it up, and then they break it down, and they go into both glycolysis and the pentose phosphate pathway. Yeah? So you could demonstrate that this molecule satisfied some of the criteria that could be a controlling resource. So the key experiment is this then. If we now prevent the synthesis of this molecule or the uptake or the utilization, we should now no longer have this organized heterogeneity. That's exactly what we did. So these are wild type cells. These are the reporters for the glycolytic state. You see they're all, of course, in this part of the colony. But now if we just block transport, this is slightly inefficient, so we'll see very, very little glycolytic cells. And if we block the utilization of trehalose, you completely lose the glycolytic cells. You still have colonies, they're still growing. They're actually much smaller. Even though we have scaled it here, you can see this is smaller than this. But we now see how you can start creating 2-state systems in a population of cells starting uniformly from one metabolic state. Okay. So the idea of the model is this. Your cells are constrained by the environment that they're in, which means if you're in a crappy low glucose environment, you can't do glycolysis, you're stuck doing gluconeogenesis. But as you do a metabolic pathway and carry it out, you start making new molecules. And as you start making new molecules, they build up, they accumulate, they've actually then changed the environment because they're being put out, right? And if they reach sufficiently high levels here, now some cells could stochastically switch to start taking up the trehalose. In this case, trehalose, but it in theory could be a variety of other molecules. The moment they do that, the amount of this molecule present outside drops. And it drops dramatically, which then means you're left with the other cells that are stuck being producers because they don't have enough of the molecule outside over there. So they're continuously producing while the other one continuously becomes a consumer. And you reach an equilibrium. Many of this we could also then model. The model would predict something like 30-70, and we saw something like 30-70, those ratios. Those are little details. But the idea is exactly this. So collectively, this is the whole process that we found in this study, where we build up a new molecule that is put out, it changes the environment, and it's perfect because this is a consumable molecule that is useful for an entirely opposite metabolic state. So now you can take it up, and then you have a stable population of producers, consumers that are working together for this collective community of cells to work as a specialized set of cells together. We'll take a couple of questions here. I wanted to stop around here. Yeah, stop here. Good. Only because the next part, we will go into lots of other gory details of pathways and how you could have whole networks functioning. But the idea is to illustrate this idea, bringing it back to basic metabolism and the two most famous pathways, glycolysis and its reverse process, gluconeogenesis. We take questions here. We can start. Yeah, we can start.
Yeah. So if I understood correctly, between the light and the dark parts, there is a difference in cell number as well?
Yes, there is. As the colony develops. So this is how it goes. I didn't include some movies and all over here. You're starting a colony here. Initially, there are very few cells. They're also separated by space. They're all gluconeogenic. Okay. As you're going, I actually should have shown that. So this is what you see. The colony starts getting bigger and more crowded. For the first few days, essentially all cells are gluconeogenic. So you see them going up like this. And around day 4, day 5, they start stabilizing in number. Okay. The first 3-4 days, you have no glycolytic cells. Around day 4, they just literally emerge. This is the shape of that curve. And then they stabilize. And the ratio approximately is about 2 is, this is about 65%, this is about 25%, and there's about 10% that are low metabolic activity or no metabolic. And this stays stable for this colony now for many, many days. We can go as long as, 25 days, 30 days, a month. They continue this way. They just become more and more reinforced in what they do.
So, given that, since initially all the cells are gluconeogenic,
Yeah.
and as they move towards the more spatial compartmentalization, per cell, do you see a change in the glucose production rates? Because
yeah, so that's a good question. How will we do that experiment? It's a good short question. How do we measure that?
Yeah, like at least could be by the same way that you have calculated the
So I'll just say
ribose
that
phosphates.
when we did this, we were the first people in the world to do this in a colony. So that's the state of the art. Okay. So now a few other labs have started doing this, but I'm just saying that's a great question. And we need to know that. One thing we can measure, which I'll talk about in the next lecture, is use transcriptome measurements, groups of cells, all of that. We can do that. And that gives you a certain type of information. We can't measure molecules at that low number. Single cell or small groups of cells, we did measure, of course, bulk, right? So we isolate light cells and dark cells. And so we know for X number of light cells, we have this much. versus X number of dark cells. And so we know relatively how much of the metabolite differences are there in the bulk. We don't really know between cells or between small groups of cells what the difference is.
Yeah, so maybe like in the initial colony when there is no light and dark differentiation, you have like you take a bulk measurement, normalize that to the number
Yeah.
of cells,
Yeah.
and then you do that same thing for just the dark cells. So that
Yeah.
might give you an idea of
Yeah.
per cell glucose production.
We have some of those numbers because we have that from reporter information. We have some reporters, right? So we have reporter level information at a much more fine resolved thing. There is reasonable difference, but so look at it this way. If you take all the gluconeogenic cells, okay, let's say they are here in terms of some gluconeogenic readout, all the glycolytic cells would be here. But you see, of course, there is this spread. They group very nicely. We'll show some of the grouping later on. But there is a clear separation somewhere here. There is this clear differentiation. But within this, there's a lot, quite a lot of noise, which is the original idea of stormtroopers versus resistant fighters. They're all individually sufficiently different, but they nicely bifurcate into groups.
And just a follow-up question, like, so essentially the dark cells are more anabolic and the light cells are more catabolic. But in the perspective of the whole colony to expand, why would you want more cells to be?
So we will discuss this on the next lecture. That was the next paper. This was the first. That was the next paper that did all of that. And then the last part is actually the overall energetics of this whole system. Okay. We haven't yet completed that part. But this is the exact obvious what we need to know. So what I've given you now is very much of a bird's eye view of saying now opposite metabolic states can exist. And they're existing in some kind of a dependent manner where at least one state entirely depends on the presence or the existence of the other state, except that they both can't happen at the same place at the same time. So they're happening in two different places, right? So that's the series of thoughts here. Okay. Yeah.
In case of low glucose, is it really because of the glucose is low or the glucose has depleted and become no glucose at a point, and that is sort of giving the phenotype of
yeah, that's a good question. That's a great question.
Gluconeogenetic
That's a great question. You can imagine that in all our discussions, this was extensively discussed. I don't have an answer to that. Partly because experimentally, and this is also why I'm saying there's this challenge of some experiments that just aren't obviously possible. We haven't yet come up with a good way to actually test that, because our limits of resolution are still limited by some amount of space. So we don't know. We don't know exactly what is the trigger. There's also another question in here. See, I'm saying now all cells have equal opportunity to access trailose and switch. Which one switch? We don't know that. We know that they switch, and after that we can explain everything, and then if we block that switching, they don't switch and all of that. But we don't know which cells switch. So it's entirely possible, coming back to your question, that internally there's a difference in the relative amounts of the reserve. And maybe it's either only these that switch or only these that switch. I would intuitively guess that the ones closest to this are the ones that switch, right? Just if they consume a bit of trailose. So they have, this one has less trailose than this, but it has enough, let's say. But by just some stochasticity, this consumes a little more trailose that is taken in. It drops down here. Then it's a continuous forward cycle, because once you start glycolysis, you're going, you're not stopping, which means then it is driven downwards this way. And so there's a constant driving forward and a pulling back that's going on over here. It has to be that way. This is, of course, just by logical inference. I don't have data for this. But if we had an answer to your question while looking at this and had a way to follow them in real time over time as they developed in a colony, then we could answer that question. Right now, we don't have a way to do both. But that is the question.
I sort of have a thought for his question. So is it possible to mix the yeast cells in a way and grow them such that they do not form colonies? And how can we do that? Is sort of make a suspension in an agar solution,
agar,
yeah,
loose agar.
glucose, yes,
Yeah.
agar, and then the soft agar colony in cancer cells that we do, we
Yeah.
basically do not let the cells come together. So if we do the same for the yeast cells and let them grow individually, they will still form a few
Very, very good. Very good question. So what happens if you change the properties of their uh, uh, the place they're growing in, right? For this kind of two-state system, you need a solid system. And in the beginning of the next class, we'll tell you what advantage it gives to this community. Now, if you make the substrate soft, or if you go into liquid cultures, we also see in liquid cultures these metabolic specializations of parts of cells. That's actually the famous yeast metabolic cycle, where different cells, they kind of start taking different shapes and metabolically oscillate. But there the difference is now, you're no longer restricted by space, which means you can't really create little micro gradients of the resource. If you're more well mixed, essentially the resource is fully diffused and you don't have slight gradients that form. So there is that component of gradients of resource driving this. Of course, we don't know what it is right now. So the phenomenon changes, but you still start getting it. But now you see, if you're in a well-mixed system, how do you create a stable exchange system between populations of cells? Because everybody is everywhere, right? So that is part of the reason why you need spatial constraints. It's a question way back.
So when you say they switch, do they also switch back?
Yeah, they can switch back.
And
fact,
does
we
this
know that they-
happen before they reach an equilibrium or something?
No, so there's a constantly shifting equilibrium, because what we also know is that the cell number is constantly increasing here, right? As the colony is growing, the cell numbers are constantly increasing. So it's actually a shifting equilibrium. Let's say you have gluconeogenic and glycolytic here, right? So this colony is expanding and moving. So some of these become more gluconeogenic a little bit or stay trapped there. But many of these over here and over here, they switch back. So the light becomes dark. And some amount of dark has to be becoming light because of the nature of the expansion. We don't have the answer to that because we can't follow each of these cells in this kind of a colony that's growing. This is the kind of stuff that you would want to do with lineage tracing. That's what mammalian people do, right? They kind of mark a cell and then they follow the cell. We don't have that here. In theory, we can do it, but then we'll have to find a way to invest in the ways to look at this whole phenomenon. We don't quite have that yet.
So maybe a follow-up to this question that I wonder about. So right, can you measure the level basically of Trellos at which cells would switch?
Yeah, really good.
And then kind of is there high hysteresis of the levels? Because right, if you basically have two switching levels, then even in a well-mixed population, you could actually maintain two subpopulations of cells.
Yeah, so we want to do that. Right now, we don't have a way to do that at each cell level. We only have these slightly bulk readouts using mass spectrometry. There are no other internal ways to quantitatively know this difference. That's a really good question, because that's at the heart of what is at the stage of switching. That's really what we want to get to. Right now, we don't have a way to do this. So we need to make some kind of tools, chemical tools, that will allow us to do it. So we're thinking along the lines of metabolite sensors. I haven't gone into that at all. Visual reporters of metabolic key metabolites. There are very few that exist. They're actually very difficult to make for a variety of reasons. But that is a really important space. If we have those, forget these cells, right? You would then start looking at it in tissues as they differentiate and so on. And that's really a holy grail. That's really a holy grail. But in this case, yeah, that would be very useful. We want to do that. We can't do that experimentally.
One question in the back.
Some questions.
So I'm thinking about a different system here. I'm thinking about a tumor, and inside that, I'm guessing that there is a metabolic heterogeneity there as well.
Yeah.
So my question is that heterogeneity like truly random, or is there an organization to that as well?
So that was kind of the philosophy of the question actually that we were addressing. How much of it is random versus how much of it requires is organization. Based on this, there's tremendous value in having organization. And if I complete my next lecture, you'll see why it's so useful to have organization. In a tumor, I don't know, but it's very clear. We know in tumors, for example, that cells are constantly exchanging stuff. That information is there. And we know that cells inside a tumor help the cells outside a tumor to grow versus, the other way around and so on. What has not been addressed in that context is the need for specific types of organizations and pattern formings. Now, it may not be the case that they're so predictable and so nice and they form. This is a simple system. This is a toy system. Essentially, we're looking at two cell states, right? Whenever you go to a tumor or an organism or an embryo or something, you don't have two states. You're dealing with many, many more states. It's just nice to break down the problem to this basic. But I would really predict or guess that there is this need for spatial organization as you are developing, differentiating, and metastasizing. Question here? One. Go for it.
Back to the tumor. I mean, tumors also form blood vessels, right?
That's right.
So then they do have a higher degree of spatial organization. Can we say that? Okay.
Yeah, they do. Blood vessels are important. And what we found, unpublished, is that actually what happens here now, as you develop even more, is that these dark regions, what look like these networks, they start forming hollows inside and they're filled with liquid. So actually, these colonies are finding ways to transport or move nutrients across the whole system. Yeah, it's there. But you see that that is another great way to get more. So with blood, you get lots of wonderful things, oxygen also, nutrients, and that's another nice way to get nutrients to different places and share. So yes.
And my original question. So coming to my question is, when you say in bright field, you see dark and light cells.
Yeah.
Are they really dark and light cells? Or
No,
is it
not
just
really. I mean, there are two parts to it. One is that the cell densities.
Yeah.
Let me go back here. Because it's it's three-dimensional. These colonies, these are from the top, they look like this, but actually from the side, like if you look at it from the side, they kind of, you have cells here. This is the, let's say this is the media, the agar. Then they grow kind of like this. And so obviously with light penetration, some of these thicker regions are dark. But the second thing is the gluconeogenic cells, they not only have lots of trellos, they also have lots of things like glycogen and so on. And that changes the optical property of the cell.
Okay.
The glycolytic cells don't have much of those molecules, and so they're actually much more permissive to visible light.
So let me put a hypothesis and you
both
can
are
tell me if this is correct. So there is stochasticity in the growth rates of cells.
Yes.
So it is possible that some cells just grow very fast, and because it is a depleted medium, it forces, when the density becomes above critical one, it forces the cells to switch states.
Yeah, it also comes back to this. Presumably, and this is something we've thought about, somebody just has to do this, where we engineer growth-related reporters. We've done a bit of that. So the absolute best indicator of growth is ribosomal biogenesis. Okay, so that's a gold standard indicator of growth. More ribosomes, assumption, more growth, right? So your signature of a growth state. If we now engineer a ribosomal reporter in these cells and quantify differences in amounts of it, we'll get an idea of differences in relative growth. There's a way, I mean, in theory, it can be done. And now if we get to that, then we will be able to answer this kind of a question. How much of a relative differences in growth are there? So at bulk, you see that these grow slower and these grow faster. And that I'll show you in my next lecture. But fine-tuning this, which ones switch between here and here, might come down to slight differences in growth rate and their ability to consume a resource. A very nice kind of a way to think about it. Okay. One more question.
I can ask the question later because.
That's fine. I mean,
okay.
So we'll take this as the last question. Is that okay?
Yes, thank you. So I'm still framing the question right. You said that there has to be a threshold value level of trehalose for the switch to happen.
Yeah.
So why can't the two metabolic states exist together without the segregation? Is it because that the environment has to be separate for these two states to exist?
It's because of the energetics of the pathway, right? When you're going this way, your Free energy is driving you to go this way. When you're going this way, you can't reverse this, right? It's like a car trying to accelerate and shift it to reverse.
No, not inside a cell. I mean, why can't the two different kinds of cell stay together, homogeneously mixed up? Why the segregation?
So I got your question. Why can't they, in a well-mixed system, they can stay. In fact, you see that in well-mixed cultures in yeast, in low glucose, they spontaneously start oscillating in metabolism, and you see the two states present together. The only difference is you can't have a spatial organization when that happens. You just no longer, I mean, this is a very well organized system, which means you're creating gradients of resources and so on. They can stay, but then that is purely stochastic. Any cell can switch at any time based on their consumption and utilization, and that happens in your liquid culture. Here, you also have this added component of space and expansion and structure and organization.
So this is in a way more beneficial for the species to organize spatially in this manner?
That I don't know, but we clearly see a lot of advantages to having this in the colony compared to cells that don't. We see that, but we of course quantify it in a very limited, narrow experimental range. How useful one is versus the other, I don't know.
And the trevulose is being produced by the gluconeogenic cells.
Yeah, all of these cells.
The inside ones, but it's not being utilized by them. It has to travel outside.
Yeah, it is available outside. I mean, there's a slightly good question here, and how do they get out? I actually don't know. We don't know, but it's there. There's lots of it there, and this build-up is important.
One last thing.
But in a way, there's a separate thing here. There is some extracellular cue, because if they just build it up inside, they don't switch. They never switch. They just build it up. It has to come outside, and then an outside signal is being transmitted. We don't have the answer to that.
Right. And this, you said the colony matured over 6-7 days.
Yes, these are 6-day or 7-day-old colonies.
But given that there's limited glucose in the medium, eventually they will all die, turn gluconeogenic first,
they
and
don't.
then
They turn gluconeogenic, but they only don't have much glucose. They have lots of other stuff, and we'll get into that. Actually, we'll get into all of that in the next class. How you go into different pathways from other sources and how important that can be in creating a self-organized system like this. Essentially, what I've kind of done here is taken you from large self-organization to specific molecules. That's it. It's at the overview level. But yeah, no glucose, but lots of other things are there, and they don't run out for a long time. The plate dries before the nutrients runs out.
Okay.
Okay.
So let's thank
very good.
Sunil for
Thank you.
the lecture.